Good afternoon, everyone. As I was introduced, my name is Philiswa Nomgongo, and I'm from UJ. So what I want to share with you today is how can we apply nanotechnology in environmental analytical chemistry or in analytical chemistry. So if we have analytical chemists in the room, you know that first thing that you see everything as, I wonder what is happening here. And then the next thing you think, if I want to know what is happening, how can I get the sample there? Am I allowed to get the sample first? Or sometimes you find ways to find that sample because there was something that you needed to know. For instance, if you look here, it's fuzzy, you are wondering what is happening in that place. You wonder, how is the air quality? How is the water quality? How is the soil? And these things are connected. And then the next thing, like, I wish to get it. Then you get it. You come to the lab now. You want to know what is in there exactly. And all these steps, that's a step that we take as an analytical chemist. Okay, we take all these steps. They look very simple here and very short, but this part, it's easier to get the sample. But this part is the one that takes more time. Why it takes more time? Everything depends here. If you mess up here, this guy will give you a value. And that value is either it's true or it's not true. So it's important that this part is done correctly. Remember, for us to have standards in the country, there was an analytical chemist who did analysis. Imagine if somebody said there was 10,000 iron in that water, only to find out it was only 1 ppm, because something was wrong there. So now what makes things happen here is either it's a long process that's being done, and we know if you ever done a sample preparation, you use a liquid phase and you do extraction after extraction and after extraction. Before the end of the day, you are tired. So that takes longer. So the question is, can nanotechnology help us to shorten the time of analysis, to be able to get better results and get the data we are looking for, and maybe in one day? Now, one will ask why I combine the two. We will recognize here there are two things that are combined: it's environmental chemistry and analytical chemistry. Why? Okay. First, before we know that in order for us to get certain answers, one discipline cannot give us that answer. And when we work together, we are able to get answers. For instance, I can't do social assessment because I don't have the language to talk to the people. Yes, I can get the numbers, but I can't relate them to the social. So it's important that we come together and then we work. Okay, when we work together, we are able to address many things that one subject cannot address. And then, now when you look at now, how does nanotechnology come in here, is that as there is a progress in any discipline. If you don't know, check when you were doing your first year in whatever discipline and check now, you will see that there is more progress because there is more knowledge that is being discovered. In such a way that in other areas we see some blaring. For instance, I always say there are times where I don't see, chemical engineers, I don't see the difference between the chemical engineer when we do the research and what I do, because it's actually, that's what it's supposed to do. We are addressing one problem. You come this side, I come this side. At some point, we come together and address one thing. So why this story is because if you are have interest in the environment, you will need to have an idea on the environmental issues. And if you want to know the environmental issues, you will need an analytical chemist, or you need analytical chemistry background to be able to tell the water has a lot of iron, or a lot of metals, or a lot of organics. So together they come together and give us analytical environmental chemistry. So we will know that for analytical chemistry, we involve a lot of things. Those are the things that are involved. And when you come together, when you have an analytical method and you see an environmental issue, you'll be able to solve them together and get the field that will be able to address both. Now, it means that we are able to apply analytical chemistry to solve environmental issues. So how do we do that in my group now? How does nanotechnology come in here? In this case, you can see here, these are the normal processes that are involved in analytical chemistry. We have your sample collection, you have your sample preparation, and you have your detection. And our strongest point is on the sample preparation. We also try now recently to try to find ways on how to develop the sample preparation techniques, the sample collection. Why because when you go to the field, sometimes you have to get a lot of water. So we try by all means not to have to carry a lot of water and see if we can do the on-site sampling using the nanomaterials and come and only do the detection in the lab. Now, why sample preparation? Remember I said to you, if your sample preparation is not correct, you will always get a value. That value is either is correct or is incorrect. A good example is here. You see here is your sample. Imagine this as your water sample. Water contains other things, and you have an interest. Let's say you want to detect caffeine. Now let's say the star is a caffeine there. If now you want to detect caffeine, if you take that sample and analyze it, this is what you are going to get. Can you see that now your interest is very low? So chances are you will come and say, no, here we are fine. We have a very low concentration of caffeine. But if now you do a proper sample preparation using the nanomaterials, you are able to, you are able to use, these are the sample preparation that you would develop when you are using nanotechnology. You could prepare your own SPE. Like, we buy SPE from Sigma or other shops, but you can make your own. You can make SPME, you can make magnetic solid phase extraction, you can make dispersive solid phase extraction. All of them, they have their own advantages and the disadvantages. But one will always design a method based on the needs. So once you apply those, what you are able to do now is to separate the matrix. There is a matrix separated, and be able to detect the analyte. Now you can see now, relative to what you had before you had a proper sample preparation, now you have a data that you can say, this is a trusted. Now, how do you design this? The good thing about nanomaterials is that you can design them to fit the purpose yourself. For instance, if you want a target to target a certain to be specific to a certain analyte, you will make sure that it has functional groups that respond to that. Or sometimes you make something called molecular imprinted type of materials, where you first imprint with the analyte of interest, you remove completely the analyte of interest, then you apply in your water. The only material that will be attached to the material, the only analyte that will be attached to your material is the one that you are imprinted with, meaning that you are making a cavity that will fit a certain molecule. So in that way, those things is the things that you can design them in the lab. And also you can add functional groups, or sometimes you add many templates to make sure that you don't do one analyte, but you do more. And that's what make the field to proceed in terms of using the nanomaterials. And okay, now an example that we have done in the lab. So what we've done in the lab, we've made a material called morph. And then when we make the morph, our interest was not necessarily the morph, was that morph has a very nice, it's a metal organic framework, has a very nice pores. Now, we are interested in the pores, not in the morph in itself. So the next thing is, when we make it and successfully make it, we then carbonize it. So here we wanted to make our own Enviro, you know that when you buy from Sigma, it's called Enviro something. It has carbon and other things. So we wanted something that is similar like that. This one was cheaper because the starting material, the only thing that we bought was the iron. The ligand we didn't buy, it was from the plastic. So we learned from the group here in CSIR that you can make more from the plastic. Then from there, we make a carbon, then we apply it in different way. In this case, we develop a method where we call it a step-bar solid phase extraction, where you have this thing has a step-bar, and you can pack your material and be able to stir and get the data you are looking for. So what was these are the characterization. I won't bore you with that, but I want us to look at the results. So what we then do is that we wanted to, because we carbonize at different temperatures, we then test everything that we have and see which one worked better. And then we then applied it in different modes of solid phase extraction. Remember I said when you have the nanomaterial, the best method that you can develop for yourself is solid phase extraction. So we did all this. What is the difference with this? Sometimes if, for instance, for dispersive solid phase extraction, it's good because it has a direct interaction with the water. But the challenge sometimes is how you recover the adsorbent. And then when you pack it, sometimes it clogs the frit, but now when you can pack it in such a way that it's protected and then it doesn't come out of the host, then you'll be able to get a better results. And in this case, we are looking at sulfonamides, one of the antibiotics that are widely used by both human and animals. And then in another case, we are looking at the PFAS, and then we know that everyone now is looking at do we have these forever chemicals in our water system. Here, we wanted to develop a magnetic solid phase extraction. And then characterize, we got it, and we confirm it's what we are looking for. And this is how then we develop the method. Now, the illustration here is that we have a magnetic nanoparticles, and then you have a sample. Usually all the things that we do is we apply in real water samples. Then you have a sample that contains the analyte of interest, which is the yellow, and the green is interfering ion. So here I want to show that the way the material was made, it was able to actually retain the analytes of interest, and the retention was based on electrostatic interaction and I think it was pi-pi bond interactions. Then in that case, we are able to get separation and get the proper results that we've got. And these are the results that we did, what we got. What we first did was to compare the material, the starting material, and see if what we prepared, does it perform better? Now you can see these two, they perform relatively better than one will ask, why then you proceed and make a nanocomposite if this one works so better? The challenge with this one is recovering from the solution. So this one you use, if I can go back, you simply use an external magnet. The adsorbent is here, then you do by magnetic decantation. So it keeps the adsorbent, then you decant after elution. And then it works better. Then we can say, okay, then this is working well. Now, the other one, we did a syringe, normal syringe from Discam or any shop. We packed the material and we were able to do the extraction. In this case, we were looking for parabens. Now, what I can say in all the nanomaterials or nanomaterial-based method that we developed, we were able to either sometimes do a combined type of analysis where you combine different type of pollutants, different classes of pollutants, or sometimes target a pollutant. And what is the advantage of nanomaterials is that if the analytes, they behave the same. Even if they don't belong to the same class, you are able to extract them. And one will ask, when do we then worry about selectivity? First of all, before we want to worry about selectivity, we want to know what is in our water first. Before now we go then say selectivity. Because when you look for selectivity, it's either maybe that particular pollutant is dangerous. So now we want to make sure that it's how much is it. But we cannot start by looking for dangerous when we don't even know what kind of chemicals we find in our water system. And this, sometimes the data, especially when it comes to emerging contaminants, you find that it's very rare, some data from the South African point of view. So it helps us to be able to say, okay, yes, it's there in our water system, or it's not there in our water system. And then now the question is, now that you have this is just a sample of the adsorbent we did. We did quite a number of adsorbent in the lab. So the question is, what next? Now that you know, what is it that you think in the future maybe the group can come up with? The first thing that we wanted to do, because we saw that the magnetic adsorbent, they work very better. So we can be able to attach them to something that rotates. We can attach them to something that would rotate in a way that they can now, that part can be able to be attached to whatever analyte we want in the water. So this one now is a catalog of a sort of an on-site where you go and apply the method you've done, come back and analyze in the lab. The other one is to be able to do something micro-extraction by packed sorbent, where you pack your sorbent and it's like a sort of a device that also is very useful when it comes to the extraction. It's also work on site. And the other one that we've done, now currently we are testing, is the fabric, whereby the material are protected in a sort of a fabric. Then how that's the iron is going to help us? We can take it on site, we can use it in the lab, but it's easier. You just use something to take it out from the water. Then one will ask, why such efforts? It's because in most cases when you deal with nanotechnology, people will always ask, what if it leaks to the water? So we try by all means to address even the part of leaching, so that when we go on site, we don't end up leaving all the nanomaterials and nanoparticles in water. We know that they are protected. And you also try now, because sometimes to make very good adsorbent, you need expensive chemicals. We try by all means to find alternative ways on how to make nanomaterials from what we have. Either it's waste or it can be something that is being planted. And then lastly, the goal one day is that we would like to be able to have, after we've tested whatever it is, to have an in situ detection. And in this case, this kind of a research, we would like to apply it in the rural areas, because what we have discovered in terms of our results, because we don't only do research in Joburg or in Gauteng, we go all over. We realize as much as in Joburg you would detect many pharmaceuticals, in the rivers, in the rural areas, you don't detect those, but you find a lot of microbial, because that's what you will expect, because there are cows, there are goats, there is everything. So one day we wish to have nanomaterials that will be able to respond to the microorganism. When they open the tap, if they have a tap, it will be able to tell them by color or by any other form that this water is not good for you. This part does not need you to be educated. It does not need a PhD. If you tell them when the color is pink, it means the water is not good. Trust me, they are very, very loyal. They will tell you the water was not good because that part was green or pink or yellow. So this is a part that is still really hard to do, but it's a wish that one day we'll be able to get it through nanotechnology. Then lastly, I would like to thank the sponsors and also the organizers for inviting me to share my talk with you and my family that I spend time with at UJ every day. Thank you.
Yes, but anyway, it was very interesting. But the other part that I was more like into concerning to it is the fact that you are targeting the rural in terms of the color and what. But now when you look in the industry, I'm from paper industry, I think the very same principle can be more beneficial also in manufacturing industries. Just to look at the color, you'll empower all the workforce from the beginning upwards. So thank you very much.
Thank you very much.
let's join me to again thanks our speaker. This is small token of appreciation from CSIR.
Thank you.
Thank you.